Okay.
Recording.
Well, hello to everyone. This is time for our second talk in our seminar today. I am Eduardo Serpa from Universidad Católica in Chile, and I have the pleasure of introducing our next speaker. He is Pablo Blanco from Laboratorio Nacional de Computação Científica in Brazil, LNCC. Well, Pablo got in 2003 his diploma of engineer at Universidad Nacional de Mar del Plata. See, Pablo from Argentina, no?
Yeah.
Okay, speak very well Spanish, I guess.
Yes.
And then in 2008, he got his PhD in computational modeling at LNCC, where he is a researcher, okay, since 2009. His research interests are in scientific computing with application in fluid mechanics, cardiovascular systems, and hemodynamics, studying blood flows. Yeah. For today, the title of his talk is Toward Real Life Computational Hemodynamics. So, Pablo, please go ahead.
Thank you, Eduardo, for the introduction. I also would like to thank you, Juan, for the invitation to deliver this talk. So, as already said, this work is about computational hemodynamics, that it is modeling and simulation of blood flow. This talk is not technically challenging. I will present quickly the equations behind the models that we use, but I would like to present here and to open your minds towards what is coming out there in terms of medicine, in terms of physiology, and how scientific computing and applied mathematics are embedding their strategies, their techniques into improving our knowledge about the mechanisms behind the basic physiology, the physiology of diseases, and so forth. So this is a journey that I'm going to present here, work that spans over 15 years, but we have been on this journey for over 20 years. This is at the National Laboratory for Scientific Computing, which is one of the pillar institutions of the National Institute of Science and Technology in Medicine Assisted by Scientific Computing, the MACC, which was recently renewed. So the funding was granted in the last call. So this is at the very edge between scientific computing, applied math, biomedical engineering, and medicine and physiology. And that's why the talk will focus on what we can do and where we are heading with the tools that we have at hand, coming from the engineering and from the applied math end, right? So let's start. This is not the work of a one man. So right, this is a whole team. The people that was involved in the material that I'm going to present are these two friends of mine, Lucas Müller from the University of Trento in the field of applied math and applied numerical techniques, and Gonzalo Masutalu in the biomedical engineering field, working at the University of Auckland, and a bunch of students, PhD students, master students, postdoc researchers that have contributed in one way or another to the material that I'm going to talk about next. So I would like to thank these people and also those people responsible for bringing us questions about the mechanisms underlying medical issues, right? In the fields of cardiology, in the fields of neurology, nephrology. So we have partnerships with here in Brazil with the Albert Einstein Hospital in São Paulo, with the State University of Rio de Janeiro, and people from abroad in the field of cardiology and neurology, partners in Canada and in the United States. And these people have opened a lot of doors bringing us very interesting questions and data to drive our research. So I also would like to thank these partners, without whom we wouldn't be where we are now. So when we talk about scientific computing in medicine or in biomedical engineering, we have to focus on some challenges, like understanding fundamental interactions that are currently understood through data or through hypotheses. Okay, through models we can test and we can validate, verify these hypotheses. The prediction of physiological phenomena, the analysis of the aspects related to the onset and the progress of diseases, the way in which we can diagnose pathological conditions using models, surgical planning as a way of doing playing around with the models, right? Modifying parameters and modifying simulation settings that can represent these abnormal conditions induced by man, right? Induced by the human intervention, and also the development of therapeutic strategies, also guided by scientific computing. All this within the context of patient generic, which is at the population level, or patient-specific settings. And this is where we are going these days, right? Towards a patient-specific medicine, because as we will see, establishing decisions at the population level may not be beneficial for all patients, and models can be very important in determining these specific conditions under which the cardiovascular system of each individual patient works. So this, of course, is a multi-scale problem, right? Spanning temporal and spatial scales. It's a multi-physics problem. We have solid mechanics, fluid mechanics, chemistry. So this is in a nutshell. When we are talking about modeling blood flow, we have to take into account that we have two different temporal scales and dynamics, the slow dynamic and the fast dynamic. The fast dynamic taking place at the scale of the heartbeat, and the slow dynamic taking place at the scale of months, of years, right? And within the fast scale, which is the way in which the signals occur and develop within the cardiovascular system, we have a local dynamics, which is the circulation of blood flow in specific locations of the vascular system, and the global dynamics, which is the behavior of the cardiovascular system as a whole in a more holistic manner, the arterial system, the venous system, the pulmonary circulation, the cardiac chambers. And within the local dynamics, we can go down into more refined aspects, like fluid-structure interaction between blood flow and vascular structures, blood flow and the heart, blood flow and the different organs. And within these interactions, microcontinuum analysis, which would yield information about how the tissues remodel because of the loading posed by blood flow, the loading posed by pressure, by blood pressure. So all these different scales are coupled because these signals that trigger vascular remodeling are responsible for bringing changing adaptations at the systemic level, and so we can reverse or progress some diseases because of local conditions, right? And that's why we talk about a multi-scale and multi-physics problem, right? So in this talk, I will focus on a very specific type of model which is one-dimensional blood flow models. And these one-dimensional blood flow models, in the way we are approaching them spans over the fast dynamics scale, focusing on local dynamics and on the global dynamics from the circulation in the major in the central arterial vessels towards the peripheral locations. So when we also when we want to characterize real-life computational hemodynamics, is because we have the goal of simulating conditions observed in real life, right? The cardiovascular system is a system that is not the same when you are at rest, when you are sleeping, when you are exercising, when you are with anxiety, right? So when you perform some maneuvers, when you perform some accelerations over the body, right? So you have different excitations, environmental excitations, and also intrinsic conditions that change. And this is responsible for altering a bunch of aspects, a bunch of elements within the cardiovascular system, from the mechanism, from the feedback control, like the baroreflex control, the chemoreflex control, which are mechanisms through which the cardiovascular system regulates itself to maintain a homeostatic state in terms of blood pressure, in terms of required blood flow, right? So there are many ingredients that should be considered towards what we call real-life hemodynamics. From having a high-fidelity human anatomy to describe blood flow in the different locations of the human body, modeling, as I said, feedback loop mechanisms like baroreflex control, chemoreflex control, modeling the circulation, the microcirculation, transport of oxygen, removal of carbon dioxide, the regulation of the pH, regulation of the pressure, also because of humoral mechanisms. So there are many aspects that makes this problem a very challenging one. Maybe not from the technical mathematical point of view, but from putting together sparse data, because as you can perceive, there is no enough data to accommodate these models, to refine and to tune these models. So we have to go ahead with the established knowledge and then collect data from patients to tune the models to specific situations. So this is the challenge we are facing. The first approach we took like 15 years ago was to bring together data, anatomical data from the established literature. So this is data that was already known for over 200 years. Well, I am exaggerating, but anatomical data, anatomy is the oldest domain within medicine, right? Because scientists started the field of medicine by studying anatomy. So anatomical data is the one that is there, has been there over decades, and we have collected a lot of data by reviewing the literature, and we have brought also mathematical models that describe blood flow in the formable pipes, which are the models that we are using, and in a while I will describe more in detail. And we put this together at the scale that was unprecedented so far. So in 2015, 10 years ago, we developed an anatomically detailed arterial model. And more recently, 2 years ago, we coupled the venous system. So we have a closed loop representation of the cardiovascular system consisting of the arterial side and the venous side, right? And this is basically an in silico laboratory for testing hypotheses, for conducting cardiovascular research, for performing the design of experiments, for assessing therapeutic interventions, because we can play around with the connectivity of the topological connectivity of these arterial and venous networks, right? So basically, this is what I am talking about. This is the representation. This is basically what you can understand as a physiological atlas, right? Because we have not only anatomical data, but we have simulation data on top of this network, right? This is the arterial network consisting of more than 2,000 vessels. The venous network, we are still working on this with nearly 200 vessels, and with the specific focus on the brain and on the coronary circulation in the heart. And because this is related to our current interests, and we are considering other coupled systems like the cerebrospinal fluid system, and we are also considering the interaction in detail with the cardiac chambers through specific lumped models of the ventricles and of the atria. So basically, these are the equations. I will go only over the most important ones, which are on the top left of the slide. We describe the blood flow, the one-dimensional blood flow, which is a model that you can derive from the Navier-Stokes equations. You can also, this can also be seen as a generalization of the Euler equations that were derived in the 18th century, right? So this describes, this system of equations describes the area, the lumen area of each vessel, because the vessels are deformable, the flow rate through each vessel, and the blood pressure, which is considered constant in each cross-section. So this one-dimensional models describes in a distributed manner the flow rate, the pressure, and the lumen area along each vascular segment, arterial or venous segment. So these two systems of this system of two equations, which is mass conservation, the first one, momentum conservation, the second one, are complemented with the third equation, which is the equation on the left, the third equation on the left, which is a constitutive equation relating the lumen area and the blood pressure, and it explains how vessels deform as a function of the stress that it is applied over the arterial wall, right? So this equation gives us a hyperbolic system of partial differential equations, which turns into a parabolic one when you consider viscoelasticity. Viscoelasticity is a dissipative mechanism which is present in the arterial wall. It is present in the cardiovascular system, and it is responsible. It's one of the major ingredients in dissipating energy in the cardiovascular system, in the major arteries, in the major vessels. So viscoelasticity is an ingredient that makes this problem even more challenging from the mathematical and also from the numerical point of view. So the rest of the equations basically describe what happens at junctions. Junctions are points where more than two vessels converge, and we have mass conservation at each junction, and we have total pressure conservation, which are the coupling conditions derived from Bernoulli theory. We have equations for the cardiac chambers relating pressure and volume in each one of the ventricles and atria. And we have other equations that explain how, for instance, cardiac valves open and close, the dynamics of the opening and closure of cardiac valves, how the activation functions for the cardiac chambers are. So the cardiac chambers are active elements within this partial differential equation, ordinary differential equation couple problem. The loading belongs to the activation functions provided by these functions here on the left that describe how the strain, the active strain is generated in the cardiac chambers. And then we have other closure equations for the peripheral circulation, for the pulmonary system, which are basically in some cases algebraic equations, in some cases partial differential equations that relate pressure, volume, flow rate, and provide us with a closed loop representation of the cardiovascular system. So basically we have the arterial system, which is a 1D, the venous system, which is 1D, and between these two systems, we have ordinary differential equations coupling on the heart side and on the peripheral side, which is representing the smaller vessels in the cardiovascular system. So to deal with these governing equations, we have to develop our own numerical techniques, our own strategies, because the strategies available out there were not efficient to perform simulations at the scale we were approaching. So we employed a finite volume method, which is high order in space and time between order 2 and order 3. We have local time stepping strategies where each one of the vascular segments is solved independently with a specific time step to avoid excessive numerical dissipation and to be efficient in not constraining the entire system to work at a pace restricted by the smallest one of the vessels. As I said, the viscoelasticity brings a parabolic flavor to the system of equations. So we hyperbolize the system to deal with the viscoelasticity and to approach the problem with explicit methods, which is our goal. We also treat consistently the phenomena of viscoelasticity at junctions, and this was an open problem until we started working on this, because at junctions usually people just discarded the viscoelastic phenomenon, and assumed that the vessels were elastic, but this was the cause of some artifacts at the junctions, as you can see here. Oh, sorry. As you can see here. But at the same time, we had to develop all these numerical techniques within a paradigm of distributed computing, because we are talking about thousands of vessels, and in a while you will see this scales up to hundreds of thousands of vessels. So it is very computationally demanding, even if the model is one-dimensional, and even if the model is a slight generalization of the Euler equations, of the one-dimensional Euler equations, we are taking this problem too long at extreme scales, and so it was required to develop parallel computing strategies to deal with these simulations in a timely manner. So our techniques scale up to several hundreds of cores. We are exploiting the facilities provided by the Santo Dumont supercomputer located here in Petropolis, at the National Laboratory for Scientific Computing. So we have the right tools. We have been working in this problem for over 10 years, but we developed the right set of tools to approach this problem at a scale that was not seen before in the major groups that work with one-dimensional blood flow modeling. So just for you to have an idea, before this model that consists of several thousand vessels, which have a very stringent connection with the real anatomy of a patient, the models available in the literature were spanning over 100 vessels. The major 100 vessels in the cardiovascular system were represented in this previously published arterial model. So we moved the boundaries towards a very realistic representation of the cardiovascular system. And of course, this this had to be somehow verified against the established knowledge. And when we talk about established knowledge, we have to refer to measurements available in the literature about blood pressures, about flow rates across different segments in the cardiovascular system. Here we are comparing the model predictions on the right column with some in vivo measurements on patients. And I have to say here that we didn't use this data to calibrate our model. So we calibrated our model based on our experience, based on some engineering rules, based on some measurements about some properties of the cardiovascular system, and then the refinement was improved to represent the main features of blood pressure and blood flow across the different territories in the vascular system. As you can see here, the ascending aorta, different locations in the aorta, radial artery, in the veins, superior vena cava here and here, the femoral veins, supraceliac vein, in different locations in the cerebral vessels as well, the common carotid artery, which is the major vessel that supplies the brain, the internal jugular vein, which is one of the major vessels that drains the blood flow from the brain, the superior temporal artery, vertebral artery, which is a vessel in the head, so in the neck. So having established a verification, let's say, it is hard to say that this is a validation, because we didn't have the exact system we were representing to compare with, but we are verifying that the model predictions match the data that it is out there in the literature, right? So, of course, you can also look at specific cardiac indexes, cardiovascular indexes, which are many times reported in medical reports. When you perform an analysis, or when you perform a Nico Doppler, for instance, you can see which is your stroke volume, your ejection fractions, your coupling ratio. These are quantities that are clinically relevant for diagnosing a specific condition. For instance, a patient with heart failure has a specific coupling ratio that has nothing to do with this baseline healthy subject, right? So we can use this, we can match these indexes with specific conditions from the literature, with different physiologically healthy and pathophysiological conditions from the literature, right? Here are many indexes, all of which can be related to metrics available in the clinic. And this is what we can see, we can analyze, that cannot be analyzed in vivo, right? We cannot analyze in vivo how the pressure, how the flow rate propagate across the entire body, but we can do this with the models, of course. So what you see here is the blood pressure on the arterial side, the venous pressure on the right, and this is a simulation of what happens with the blood pressure in one heartbeat. On the left, we have the brain blood flow, the arterial blood flow in red, and the drainage of the blood flow in blue, how this happens within a heartbeat. So, okay. So, but what about other model predictions? Well, we tested the model on many different conditions, some of which I'm going to present here. For instance, the model is especially suitable for situations where we have to involve many complex mechanisms that drive blood flow. For instance, this is one complex situation that happens when you have some condition called subclavian steal syndrome. And this happens when you have some blockage on the subclavian artery, which is the major artery of the arm, right? And what happens in those cases, patients that exercise, for instance, with the arm, they exercise and they have symptoms of dizziness, they have symptoms, they can faint, they can feel transient ischemic episodes in the brain. because blood is not reaching the arm through the major artery because it is blocked, but the blood reaches the arm through the brain. And this is it's a very complex situation because the blood flow, instead of going directly through the major vessel to the arm, it comes up to the head through the vessels of the neck, and then through the circle of Willis, it goes down through the posterior neck vessels, which are the vertebral arteries, and then these vessels are connected to a distal portion of the subclavian artery, and therefore the blood reaches the arm going through the circle of Willis. In doing that, the arm can request more blood than this pathway can provide, and therefore the brain can, for some instance, receive less blood than the one that the brain requires, that is suitable for the brain. And in those cases is where you have symptoms of dizziness, or, as I said, you can faint because your pressure in the brain drops significantly. And this can be explained exactly with this model, and we compare this with data from the literature. For instance, in this case, we are comparing the predictions on the top row with measurements in patients on the bottom row, and you can see that the qualitatively and quantitatively the predictions are in very close agreement with the data available in the literature. Again, we didn't use that data to calibrate our model. So the model these are blind predictions made just by introducing the blockages, introducing the reconfiguration of the vascular topology, and this was the outcome. So as you can see, the model responds pretty consistently, even in this complex situation. But one of the challenges of this is that the model is very expensive. And this is what I said, we have to deal with one-dimensional equations in thousands of vessels. And this is computationally demanding. So we started developing a hybrid approach where we reduce the dimension of some portions of this model. And instead of a one-dimensional model, a distributed parameter model, we reduce this to a lumped model where we describe pressure and flow rate, but we reduce the features of the one-dimensional model to resistances, compliances, and inertances, which are the electric analog elements that describe the behavior of the blood flow within vascular segments. So these are the linearized equations. These are actually non-linear equations, but after a first linearization, you come up with these equations where the properties depend on the variables, and we can describe in a more efficient manner the blood flow in these segments by using these compartmental representations. The question is, okay, where we can safely reduce the dimension of these models, and we have investigated different criteria to turn the one-dimensional representation into a zero-dimensional representation. And these criteria could be vessel length, vessel diameter, vessel volume, vessel compliance, the region of the different vascular territories, the Reynolds number. So with these different criteria, we investigated how the effectiveness in the prediction of the flow rate and the pressure changed with the threshold used to transition from a zero-dimensional model into a one-dimensional one, right? So the numerical machinery is also had to be adapted to deal with this coupling between the ordinary differential equations and the partial differential equations. And we explore both implicit and explicit coupling strategies. And okay, I sorry that I had here I had a result that was, okay, disappeared. But basically, there is a sweet spot where we can turn the one-dimensional model into a zero-dimensional model using as criteria the compliance of the vessels, and this gives us a strategy to make these simulations more efficient. Now, moving again over the physiological modeling, we wanted to investigate the effect of the respiration, and this is the basis for simulating also the transport of oxygen and carbon dioxide, and we are working on that currently. So, but respiration was the first step towards that goal. So, breathing is a very important, is a semi-voluntary action, right? So you have a partially you can control your respiration, and partially it is a reflex that you breathe, right? So the changes in how you breathe affect the cardiovascular performance, and this is what we want to explore. This is well established in the physiological literature, but no one had studied the effect of the respiration on the pressure and flow rate signals, the waveforms, in the way we did here. So we added a very simplistic representation of the pulmonary of the lung mechanics. So this is here we have the muscular respiration, the muscular pressure, that it is coupled to the intrathoracic pressure. We have eventually the effect of a ventilation at the mouth, right? So these are the signals that are responsible for introducing additional effects on the circulatory system, right? So this is lung mechanics, and these signals are one-way coupled. There is no way back because there is no feedback so far. But these signals are one-way coupled with the blood flow phenomenon, right? So here we have to determine which parts of the body are coupled in the thoracic region, in the abdominal region, right? The arteries and the veins, which are described here, and these different regions are coupled to different levels of pressures imposed by the respiratory episode, right? So basically, we have two regions: the pleural pressure applied over the thoracic vessels, and the intra-abdominal pressure applied over the abdominal vessels. As you can see, the intra-abdominal pressure is positive, and the intrathoracic pressure is negative. So you have here a pumping effect, because the positive pressure over the vein, over the inferior vena cava, which is the largest vessel that you see there, this brings a very important effect on the venous return. The venous return is one of the, is a very fundamental aspect related to how much blood the heart can pump into the system, right? If your venous return is good, then you can pump sufficient blood to the entire system, and you can release the, let's say, the load over the heart, because you have a good venous return. So respiration can help you with this pumping effect. And that's why it's so important to control respiration during exercise, or to control respiration in acute situations like anxiety situations, like panic attacks, because respiration has a very strong effect on the blood flow pumped by the heart. And even if it is not modeled in this case, there are reflex mechanisms that are also affected by the respiration. So that's where we are heading, and that's why this was so important. But this pumping effect, this additional pumping effect brought by the respiration, required reparametrization of the model. So the redefinition of the parameters, mainly the contractilities of the heart. And this was based on a sensitivity analysis. So we considered different scenarios that we were going to compare: the original parameterization of the arteriovenous model. Then we considered the system when the respiration was turned on and the parameters were the original ones. And then when the respiration was turned on, and we reparameterized the model. So, and I am going to focus on these cardiac indexes, clinically relevant cardiac indexes that I talked about earlier. So these indexes are the ones that are reported here when we had the respiration turned off, right? These parameters were all within the physiological range. So if you look at these parameters are good representations of a healthy subject. And this is with the respiration turned off, right? When we turned on the respiration, we observed that some of these parameters went crazy. So they were out of bounds. For instance, the cardiac output, which is the amount of blood pumped into the system at each minute, moved from 5.8 to 7.5. So increased by more than 20%, right? And this was only due to the respiration. And with the increase in the cardiac output, the blood pressure increased, and all other cardiac indexes moved along together consistently. So, but therefore, these indexes were not clinically within physiological range, and therefore there was a need for the reparametrization just to bring back these indexes into a healthy range of variation. So this explains how respiration is important in determining the true parameters of the cardiovascular system. Otherwise, we are talking about apparent parameters that somehow take into account the combined effect of the true parameter and the respiratory effect. So here, these are some signals, some cardiovascular signals in different vessels like common carotid, abdominal aorta, inferior and superior vena cava. The blue curves are with the respiration off. When you turned on the respiration, then many of these signals suffer significant changes. For instance, here the pressure increases like 30 millimeters of mercury. 30 millimeters of mercury is a crazy change in blood pressure. So you go from a healthy subject to a severe hypertensive subject with 30 millimeters of mercury, for instance. But then when we reparameterize, we bring back the signals to the corresponding to healthy values. The same here for the cardiac pressure waveforms, pressure in the pulmonary system, the flow across the cardiac valves. The same investigation can be carried out. The same understanding can be gained. You can see here, for instance, this is a very, very clear effect of the respiration. We have inspiration during one phase. So here we are looking at 5 cardiac beats. And in 5 cardiac beats, we place one breath cycle, one breathing cycle. So we have inspiration and we have expiration. And the inspiratory phase and in the expiratory phase, we have significant changes in the quantities. And we can here quantitatively and qualitatively analyze how these changes take place all over the cardiovascular system. As I said, with a degree of detail that was unprecedented so far. So now moving into other portion of the cardiovascular system, which is the peripheral circulation, we want also to analyze what happens with the blood flow in the smaller vessels of the cardiovascular system. But our model didn't have, at the time, these vessels into their representation with a degree of detail that we wanted. So we developed algorithms to vascularize, to synthetically vascularize domains, vascular domains, starting from some feeding artery. So if you have a blue feeding artery, like in this case, and you have two different vascular territories that you want to vascularize, these algorithms are responsible for filling with vessels these regions, these vascular regions, such that the resulting vascular network features a statistical statistical features comparable to what has been observed and measured in the literature with cast models. So these cast models are post-mortem models. So they retrieve a portion of the cardiovascular system, they inject some polymers, and they corrode these models to remain with the arterial or with the venous networks of specific portions of the body, right? Of the coronary vessels, of the kidneys, of the liver, and this for humans and for animal models. So there are statistical features that these vascular networks have to comply with to be able to supply in a uniform manner with oxygen or the totality of the vascular territory. So this algorithm takes that into account, and in particular, we developed a parallel version of this algorithm to be able to vascularize complex and large regions like the kidney, for instance. The kidney, the microcirculation in the kidney, where glomeruli, which are the basic unit of filtration in the kidney, are responsible for the filtration and therefore for the function, for the renal function in the human body, right? So this is a real micro-CT photo image of glomeruli. As you can see here, there are many glomeruli. We have 1 million glomeruli in an average person. So this is a very, very large scale problem. And the idea was to create a vascular representation of the human kidney, right? So this is what we built. We built this model where you have these pyramids in dark gray. These pyramids is where the concentration of the urine goes, and this urine is collected by the calyces. The calyces are the structures in light blue, right? And therefore you have some regions that are not vascularized, I mean, are not, let's say, are not where nephrons are the structures attached to the glomeruli. So nephrons are located over the cortex. The cortex is where the light gray area is shown in that geometry. So this is a very complex structure, and the algorithm is able to vascularize this structure in a uniform manner to represent the kidney vasculature. And we took this to the extreme where we have 200,000 vessels, which is 200,000 vessels is the model, the most vascularized model, which is here over the left. And we simulated blood flow and pressure on this vascular network. So we didn't reach yet the scale of 1 million terminal vessels. We reached 200,000. And this small jump of a factor of 5 is not a minor thing, because that could imply in several weeks of calculation to develop the vascularization, but we are working towards that. So these are some examples of the pressure distribution and the flow rate distribution in the different vessels of the kidney. And these data could be used as input data to the glomeruli structure, to these glomeruli structures where filtration occurs. And we are developing models of the nephrons and of the proximal convoluted tubule, which is the first portion of the nephron where most of the blood is reabsorbed and where the urine is mostly morphed into their final feature. So we are studying renal physiology from the macrocirculatory scale down to the scale of the nephron. This is one of the lines of research we are conducting here these days. We have a PhD student working on that. And on a similar line, we have the same kind of interest in what happens in the brain. So with the brain, we were brought a very interesting question about the etiology of white matter intensity. White matter intensities are small white spots shown in magnetic resonance images. And these white spots are thought to be the cause of well, there is no causal relation, but are related to neurodegenerative diseases, to strokes. And the location of these white spots depends, depending on the location of these white spots, the diagnosis is different. But basically, the etiology, which is the cause of these white spots, was hypothesized to be the same, which is degeneration of the vascular vessels and a rupture of the small vessels that led to these white spots in the medical images. So the idea was to explore from the mechanical point of view, which was the level of pressure in the different regions of the brain, right? So we have basically two different types of vessels here, vessels at the base of the brain. We supply what is called the centroencephalon, which is here the blue region, and the vessels that supply the cortical part of the brain, which is the dark pink region here, which is the cortex, right? Our thinking brain. So we have the more primitive brain, the base of the brain. Our thinking brain is the outer region of the brain. And basically the idea was to investigate the blood pressure and the blood flow to try to answer the question about the origin of what is called the small vessel disease, which is this degeneration of the small vessels that was manifested in medical images through these white spots that I mentioned earlier. So basically, we wanted to investigate. The hypothesis was that there is a strong pressure gradient between the vessels at the base of the brain and the vessels over the cortex. Even if the brain is a small structure of over, let's say, 10, 15 centimeters long, right? So the pressure gradients should be important. And so I will speed up here. So we differentiated the vessels at the base of the brain with the vessels over the cortex. This is just images from a monkey specimen, right? So just to try to understand which is the structure, the vascular structure of these vessels over the cortex. And this is what we have in our model at the time we did this investigation. That was in 2016, 2017. We have the cortical vessels, the vessels supplying the cortex, and the vessels at the base of the brain that are these small vessels that arise from the circle of Willis. And at the time, we developed these small vascularizations, small spherical vascularizations, and we couple these spherical vascularizations at the base of the brain and over the cortex. So at the base of the brain, at the base of the brain, as you can see here, at the base of the brain, what we observed is that the pressures were significantly higher than the vessels over the cortex, even if we are talking about vessels of the same size. And that was a major breakthrough. So if we take 40 micron vessels or 200 micron vessels at the base of the brain, this LSA, lenticulostriate arteries, is this at the base of the brain. Or if we take the vessels over the posterior parietal branch of the middle cerebral artery, which is this over the cortex. So these corresponding vessels reached very different levels of blood pressure. That would tell us that the cost of degeneration of the vessels is not the same, because while vessels at the base of the brain are exposed to very, very high pressure, and they may rupture because of high pressure, the vessels over the cortex, even in hypertensive subjects, the pressure was cushioned, was significantly reduced by the entire vasculature towards the convexity. So in some sense, our cortex brings a cushioning effect, a reduction in the pressure that it is very beneficial for the vessels over the cortical region. But on the other side, this is dangerous, because if the pressure drops so much over the cortical vessels, then if you take some antihypertensive medication, then you could lower the pressure over the cortex more than you should. And this could be very detrimental in mid and long term, in terms of mid and long term outcomes. So we wanted to take this further, and we wanted to make this more realistic. So we took the geometry of a real human brain, where we accommodated the arteries of our arterial model. We split this brain. We took only the left hemisphere. We split this into the major anatomical regions, and we vascularized using the same algorithms I presented before. We vascularized all these, the gray matter, not the gray matter, but the surface that supplies the gray matter. So this is the PL surface. So the PL surface is a very complex geometry where you have these sulci and gyri, which are the different valleys and hills that the PL surface features here, that you can see here. So the vessels run through these valleys and hills of the brain, and then penetrate into the gray matter. And this is what we have built. We built the model of the PL circulation, which is the vascular network over these valleys and hills before penetrating. So these are vessels that run from the major vessels of the brain, the middle cerebral artery, anterior cerebral artery, and posterior cerebral artery, towards 40 or 50 micron vessels. That is where the columnar arborizations start to penetrate into the gray matter. Right here, I am showing this picture shows the simulation using one-dimensional modeling. So this is a simulation of the pressure, which are, in this case, you can see how the pressure in the subject, which is healthy, and in a subject that it is hypertensive, how the pressure in the brain behaves in the different regions of the brain. So we can observe large pressure gradients between the major vessels of the brain at the base of the brain and over the cortex, particularly over the posterior part of the brain, right? And in here we just sample different locations to show how the pressure and the blood flow look like in these different locations. And we perform these simulations on a healthy setting and on a hypertensive setting. So these kind of simulations can tell us how important is the consideration of defining a nice cerebral blood pressure for each specific patient. And this is the major lesson that we have learned from here. The cerebral blood pressure is very variable compared to the pressure that you measure on the arm. So if you measure your pressure on the arm and your pressure is, I don't know, 913, right? That is 90 millimeters of mercury at diastole and 130 millimeters of mercury at systole. This tells you little about the pressure in the brain. And lowering the pressure in your arm is not a necessary condition or it is not necessarily the best strategy to deal with the pressure in your brain. So there is an urgent need to come up with methods in the clinic to assess cerebral blood pressure at the patient-specific level and determine what it is a nice blood pressure for each specific patient. Just currently, the pressure, the thresholds are established at the populational level, but we are moving towards a more patient-specific definition of these thresholds. So what we observed, and this is just to wrap up here, pressure gradients were huge across different regions of the brain. The flow rate pulsatility was also very heterogeneous, and this tells us about the fatigue that the vascular vessels experience from the interaction with the blood flow. The lower pressures were located over the occipital lobe. This was an observation we made from the simulations. And of course, what happens when you have obstructions in some vessels? When you have a stroke, which is the extension of the damage, the region affected by the stroke, right? Or what happens if you have excessively low pressure levels because of medication or because of some exercise, or because you are sleeping and pressure drops while you sleep. So there are many questions that we are trying to address, but one of the major concerns currently is to try to improve the estimate of a patient-specific cerebral blood pressure from data of a specific patients. So I will finish here and just make this the message. We have some interesting databases which are publicly available. I have to check because this was the server was down a couple of weeks before, but it was up after we knew that. So this Adam Web database is there for anyone who wants to query pressure and flow rate waveforms across the entire body. This is the bibliography that I used to make this presentation. This is basically our work in the domain of one-dimensional blood flow model. So thank you once again, Juan, and the entire audience. I'm open for questions.
Thank you, Pablo. Thank you for this talk. So now we go to the question. If there are any, please just open your mic and ask yourself the question. Just to start, I have a couple of them. Pablo, you mentioned something when you talk about the respiration, but is part of the slow dynamics, the respiration?
No, well, I divide it into two dynamics. I would put this on the slow dynamics because it's at the scale of the seconds, right? But so respiration takes you perform one breath per 5 seconds or 10 seconds. So it is the fast dynamics still. Control mechanisms, depending on the control mechanism, you are between short-term control mechanisms like battle reflex in a matter of 30 seconds, chemoreflex, which is a reflex that acts over the spanning 1 hour, for instance, and then you have longer adaptations that take over months or years. So these control mechanisms are in the mid-range of the time scale, but the breathing, the respiration, I would assign it to the fast dynamics box.
Okay, and here you focus on the 1D dynamics, no, right? For the vessel and so on. But for the heart, what do you do?
For the heart, we use compartmental models. So ordinary differential equations, because we just want to model the relation between pressure and volume. we don't model the complexity, the three-dimensional complexity of the blood flow in the heart, because our interest is not in how, for instance, in the regional behavior of the myocardial wall. That would be another different line of research focusing on the heart. We just use a very simple representation, which is non-linear. I mean, it has its complexities, but it is an ordinary differential equation that relates pressure and volume.
Okay, okay. Well, if there are any question, please just ask it. I have another one, just in case. When you talk about the brain in the last part of the talk, I was thinking about you mentioned MRI, right?
Yeah.
What is important today is, or a new challenge is trying to do quantitative MRI, no? So I think that maybe your approach can be mixed and maybe go in that direction of maybe some more quantitative MRI, no? I don't know if it's far from what you are thinking about.
Well, actually, similar model, simpler models than the one I presented here are used to be integrated with quantitative MRI to try to quantify the signals arising in MRI, like blood flow, and to analyze the impact of strokes and the impact of obstructions, as well as to understand other aspects of the circulation. But in this case, we're modeling the blood flow. We could simulate the signals that you see in quantitative MRI when they characterize regional blood flow. We could do that. But the idea, at least that several groups have out there, is to couple this kind of representation with MRI to improve the predictions, the information that the MRI gives you. So there is another imaging technique called arterial spin labeling. This arterial spin labeling is more advanced. It's just for the brain so far, but this is a magnetic resonance-based imaging technique, and this gives you the blood flow, the regional blood flow, with a degree of detail which is very, very high. So this could be either used to calibrate our models, right, in the first place, and then with this kind of calibration, we could perform higher resolution simulations to refine how the blood flow behaves at the scales that are not observed in the magnetic resonance imaging, because magnetic resonance is limited by the resolution, right? But you can integrate with this, let's say, data from MRI, and then refine the calculations and go further in the characterization of the blood flow in the brain. But we don't have collaborations with people that holds this kind of data. So this is why we are not approaching that line of research.
Okay, thank you. Very, very nice, very interesting. Well, if there are other questions, please, or yeah, well, if not, we can thank Pablo again. Thank you very much, Pablo.
Thank you, Pablo.
Thank you.
Yes, interesting talk.
And